in order to ensure that all the different types of enzymes of our body carry out their function at the proper location and at the proper time, our body must be able to continuously monitor and regulate the activity of enzymes. And so far, we discussed 3 different mechanisms that our body uses to regulate the activity of enzymes. So we discussed allosteric regulation, we discussed covalent modification, and proteolytic activation. Now we're going to discuss yet another mechanism that our body uses. And this mechanism involves using different forms of the same type of enzyme to carry out a single specific type of reaction that basically takes place at different locations and different times in our body. And these different forms of the same type of enzyme are known as isozymes or isoenzymes. So isozymes generally allow our body to regulate the same type of biochemical reaction that takes place at different locations and sometimes at different times. Now, before we actually look at a specific example of isoenzymes, let's generalize what we know about isozymes or isoenzymes. So, once again, isozymes are these different forms of the same type of enzyme. And these isozymes come from different genes. And what that means is they're going to have different amino acid sequences and different 3-dimensional structures, but they will catalyze the same type of biochemical reaction. So we see that isozymes arise from different genes. They have different sequences of amino acids, and what that means is they will generally have different biochemical properties. And this is exactly what allows us to actually separate or purify mixtures of isozymes. Now, not only that, but isozymes also generally have different enzyme kinetics. So they differ in the Km value, the Michaelis constant, in the turnover number kcat, and they also differ in things like the Vmax, the maximum velocity at which the enzyme operates. And isozymes are also typically regulated by different types of regulatory molecules, different types of allosteric effectors. So inside our body, we have many different types of cells. And these different types of cells basically exist under slightly different conditions. Yet, all these different types of cells have to carry out identical types of reactions. For instance, to see what we mean, let's imagine we have a cardiac muscle cell and a skeletal muscle cell. So we have 2 different types of cells that are found at 2 different types of locations. So cardiac muscle cell is found in the heart, and skeletal muscle cell is basically found around the bone. Now, cardiac muscle cells are found in a rich oxygen environment, but it's the skeletal muscle cell that is found usually in a lower oxygen environment. But both of these different types of cells found under different conditions must carry out the same type of biochemical process, namely, let's say, glycolysis. So they both have to be able to metabolize glucose in the process of glycolysis. And in fact, skeletal muscle cells and cardiac muscle cells actually use isozymes in the process of glycolysis. So remember a specific type of enzyme that we spoke of previously. So we discussed lactate dehydrogenase. And we said that lactate dehydrogenase is an enzyme that is basically involved in glucose metabolism. And more specifically, what it does is it transforms pyruvate into lactic acid. It reduces pyruvate into lactic acid, and that at the same time oxidizes NADH into NAD+. And what that allows us to do is it allows us to regenerate NAD+, and so that means we can basically restart another cycle of glycolysis. So we see that lactate dehydrogenase catalyzes the reduction of pyruvate into lactic acid at the same time and oxidizes NADH into NAD+. Now, if we examine the quaternary structure of lactate dehydrogenase, LDH, we're going to see that it consists of 4 individual polypeptide chains. And inside our body, in the human body, we have 2 types of these polypeptide chains that can constitute lactate dehydrogenase. So humans have 2 isozymic chains for lactate dehydrogenase. We have the H isozyme chain and we have the M isozyme chain. And it's the H isozyme chain that is expressed predominantly in high concentration in the cardiac muscle cell. And it's the M isozyme chain that is expressed in high concentration predominantly in the skeletal muscle cell. So if we examine the lactate dehydrogenase in skeletal muscle cells, we're basically going to find a quaternary structure that consists of these 4 individual chains, where these 4 individual chains are all the M isozyme chain. So each one of these red chains is basically that M isozyme chain. On the other hand, if we study the cardiac muscle cells, we're going to see the quaternary structure of the LDH basically consists of 4 individual H isozyme chains. And these are shown in purple. So this lactate dehydrogenase, which is basically denoted with H4, where H means we have the H isozyme and 4 means we have 4 of these individual H chains. So we have this lactate dehydrogenase is made up of 4 H chains and is found in cardiac muscle cells. While the M4, which basically consists of these 4 individual M isozyme chains, makes up the LDH that predominates in skeletal muscle cells. So we see that it's this molecule, the M4, that operates at a maximum efficiency inside skeletal muscle cells, where we typically have a lower oxygen concentration. But it's this purple H4 LDH molecule that predominates in cardiac muscle cell, which operates at a maximum efficiency when there's a high concentration of oxygen, as we typically find in the heart, in the cardiac muscle cell. So these are what isoenzymes are. So isozymes or isoenzymes are these multiple different forms of the same type of enzyme that catalyzes the same type of biochemical reaction that takes place at different locations and or at different times. Now let's see how we can apply this to our body, to the field of medicine. So remember that a heart attack, also known as a myocardial infarction, is basically the condition by which we have a partial blockage of the blood flow to the heart of our body. For instance, previously we discussed that if we create some type of embolism inside the coronary artery of our heart, that will lead to a heart attack. Now, if there's a partial blockage of blood flow to the heart, that will cause damage inside the cardiac muscle of the heart. And if the cardiac muscle cell is damaged, it will begin releasing its internal components, and that includes releasing the H4 LDH molecule. Now, as the cardiac muscle is damaged, releasing the H4 LDH into the blood will increase the relative concentration of this molecule inside of the blood. And this molecule is typically not found in a specific high concentration inside our blood. So physicians can basically test for the presence of elevated H4 concentration in the blood, and this can basically help them show or help them discover the fact that this individual has experienced a myocardial infarction or a heart attack. So we see that not only can our body control and regulate enzyme activity by using these 3 different types of mechanisms, it can also use isozymes. And isozymes are these different forms of the same type of enzyme that carries out a specific type of biochemical process that takes place at different locations and at different times.